Bismillahirrahmanirrahim. Assalam-o-Alaikum. My name is Dr. Muhamma Ali and I will be teaching you chapter 4, the three-dimensional structure of proteins. Need of studying the three-dimensional structure of proteins. We already know that the proteins are big macromolecules. It has covalent backbone that is containing hundreds of covalent bonds. Because free rotation is possible around many of those bonds, that's why a protein can assume an uncountable number of conformations. Depending upon the chemical structure and the function, a protein must have a unique three-dimensional structure to perform its function. How a protein obtains the unique three-dimensional structure and how this three-dimensional structure is maintained will be taught in this chapter. Overview of protein structure. The spatial arrangement of the atoms in a protein is called its conformation. Among many of the conformation of a protein, only a few are predominated under a certain biological conditions. The conformation that a protein achieves depends upon the Gibbs free energy that must be lowest, and it should be thermodynamically stable. Protein in any of its functional folded conformation is called a native protein. For a vast majority of proteins, only a small set of structures are critical for their function. A protein's conformation is stabilized largely by the weak interactions. The stability of the protein is its tendency to maintain its native conformation. Native proteins are only marginally stable. If we talk about the folded and unfolded protein, they do have a delta G value that falls in the range of 20 to 65 kilojoule per mole. The entropy and the hydrogen bond of the protein with the solvent tends it to be unfold. For all proteins, weak interactions are especially very important in the protein folding. A covalent bond has energy in the range of 200 to 460 kilojoule per mole. And for weak interaction, this energy falls in the range of 0.4 to 30 kilojoule per mole. On a careful examination, we can see the weak interactions are very important, and among those weak interactions, hydrophobic interactions are predominating in the maintaining of folded conformation of the proteins. The hydrophobic interaction among the proteins are maintained by the hydrophobic amino acids that are leucine, isoleucine, valine, phenylalanine, and tryptophan. The peptide bond is rigid and planar. In between two alpha carbons that are present in two amino acids in a protein, we can see there are three covalent bonds: 1 bond, 2 bond, and 3 bonds. If we see 6 atoms that are somewhere here, this is atom number 1, 2, 3, 4, 5, and 6. These are always falling in one plane. They rotate together and they stay together. There is another property of the amino acids that are present in the protein. They do have an angle of rotation between the nitrogen and the alpha carbon, and this angle of rotation is called the phi. And the angle of rotation that is between the C alpha carbon and the carbon of carboxylic, this angle of rotation is called the psi angle. So because the angle between the carbon and the nitrogen, this one is double bond in nature, that's why this angle of rotation is not possible. Still, we name this angle of rotation as omega, but this is not counted in the protein structure. The peptide bond is rigid and planar. We have already seen that the 1, 2, 3, 4, 5, and 6, these 6 atoms always fall in one plane. Now we see why the bond between the carbon and nitrogen is double bond in nature. The lone pair on the nitrogen is shifted here, and this double bond is shifted on the highly electronegative oxygen, and this way this bond becomes double bond and the negative charge comes on the oxygen, and this way this charge rotates, and this double bond nature does not allow the carbon and the nitrogen bond to rotate. We can once again see that the phi is the angle of rotation between the nitrogen and the alpha carbon. If we amplify this region and we take this region here, and if someone is standing here and is watching at this structure will look like this way. And if we observe the angle of rotation psi here, we can see that the psi has a possibility of angle of rotation from minus 180 degree to plus 180 degree. So allowed values of phi and psi can be drawn on a Ramachandran plot. For example, in the case of L-alanine, if we take the angle of rotation phi on the x-axis and angle of rotation psi on the y-axis, if we draw the dots that are permissible for L-alanine on this two-dimensional graph, we get different regions that are permissible for phi and psi values. The dark blue region is highly permissible for phi and psi values, and the lighter blue is less permissible, and the lightest blue color is the least permissible for the phi and psi angles of L-alanine. And the white region are not allowed region for the phi and psi. For example, if we make a dot here, the psi angle is going to be 120 degree and the phi angle will be minus 90 degree. In the same way, we can see if the psi angle is somewhere here minus 60 degree, the phi angle will be again minus 90 degree. For all unbranched amino acid, they have the similar values of phi and psi values as does the L-alanine, but the branched chain amino acid have somewhat lower values as compared to the L-alanine. The glycine has too much free rotation and is falling everywhere on this area of Ramachandran plot, while the histidine is having the limited rotation of phi angle. The phi angle of the histidine is falling between the minus 35 to minus 85 degree. Protein secondary structure, the alpha helix. We start from the alpha helix. Our secondary structure in which dihedral angles phi and psi remains the same or nearly the same throughout the segment is called the secondary structure. The most prominent are the alpha helix and beta conformation. Another common type is the turn. We can see alpha helix here. This alpha helix, for example, if it takes one complete rotation from this line to this line, it is taking 3.6 residues in completing one complete turn, and the 5 angstrom distance is traveled by the one complete turn. This kind of distance for one complete turn and this kind of amino acid residues that are taken is the characteristic of the alpha helix. If we take a top view of the alpha helix, we can see there is some free space along the axis of the helix. This is also one of the characteristics. Some of the tightly wound helices don't have this free space. This is side view in the field model, and this is the top view once again by the ball and stick model. So the alpha helices, alanine is the most suitable amino acid, while the continuous blocks of glutamine and the same charged residues will not make the alpha helix. Even the bulky groups are not going to make the alpha helix. The alpha helix is common in protein secondary structure. In this table, we can see the specific values of phi and psi that are suitable that make the alpha helix. We can see the beta conformation, the anti-parallel beta conformation is achieved by the phi angle of minus 139 degree and psi angle of plus 135 degree. In the same way, type 1 beta turn that are predominantly found as compared to type 2 beta turn that are not predominantly found, the type 1 beta turn has a phi angle of minus 60 and psi angle of minus 30. So we can check these values on this Ramachandran plot. The alpha helix is a common protein. Here we come across a problem. The problem is that what is the length of a polypeptide with 80 amino acid residues in a single continuous alpha helix? In order to solve this distance, for example, there are 80 amino acids that are arranged in this alpha helix. For example, how long is this alpha helix containing 80 amino acids? We have a basic information that 3.6 residues have 5.4 angstrom long. So one residue will be 1.5 angstrom long and 80 residues will be equal to 120 angstrom. So the answer is that 80 residues that are present in an alpha helix are 120 angstrom long. So there are two types of alpha helices. One is right-handed alpha helix and the other one is the left-handed alpha helix. For example, if we stretch the thumb of our right hand and the fingers of our hand are curled in the turns of the helix, then it is called the right-handed helix. Otherwise the helix will be the left-handed. The right-handed helices are found predominantly in the nature. Amino acid sequence affects the stability of alpha helix. These are different amino acids arranged with respect to difference in their Gibbs free energy.
The highest the Gibbs free energy, the least is the tendency of the amino acid to form the alpha helix. For example, glycine has a Gibbs free energy of 4.6,
and if we see the alanine has the least 0 Gibbs free energy, so it has a high tendency to make the alpha helix. Amino acid sequence affects the stability of alpha helix. Not all polypeptides can form the alpha helix. Alanine has the highest tendency to make the alpha helix. The position of the neighboring amino acid is very important while making the alpha helix. For example, glutamine residues, if are found in continuous blocks, they will not make alpha helix at a pH 7. In the same way, similarly charged amino acids are not going to make the alpha helix because they repel each other. The twist of alpha helix is also very important because at the edge of each twist the amino acids interact with each other. In an alpha helix, the positively charged amino acid must be 3 residues away from the negatively charged amino acids. In the same way, hydrophobic amino acid must be 3 residues away from another hydrophobic amino acid. We already know that the proline and the glycine has the least tendency to make the alpha helix. Proline has too much limited angle of phi that is not suitable to make the alpha helix, while the glycine has too much free rotation and disturbs the formation of alpha helix. Negatively charged amino acids are found near the end term of the alpha helix.
Amino acid sequence affects the stability of alpha helix. This is one of the alpha helix we can see. We repeat the previous concepts that one complete turn of alpha helix is taking 3.6 residues and 5.4 angstrom of the distance. This is the amino terminal of alpha helix and this is the carboxy terminal. We can see these dotted lines are showing the hydrogen bond, are the forces of attraction between the positively and the negatively charged amino acids that are present in the alpha helix, and that are 3 amino acid residues away from each other. Amino acid sequence affects the stability of the alpha helix. There are 5 types of constraints that are involved in the formation of alpha helix. One is the intrinsic property of the amino acids. For example, the bulky groups and some of the amino acids that have rigid rotation, they will not make the alpha helix. And some of the interaction of the amino acids are also important. For example, the amino acids that form 3 or 4 residues away are the similarly charged and they are not oppositely charged. They will not make the alpha helix. Sometimes the bulkiness of the groups is involved. They are taking too much space and they do not favor the formation of helices in the alpha helix formation. Occurrence of proline and glycine is very important. Proline is too much rigid for the formation of alpha helix and glycine has too much free rotation. And also the interaction of the ends of the alpha helices are very important. For example, the amino terminal is containing negatively charged amino acid while the carboxy terminal is containing the positively charged amino acids. So the secondary structure is the beta conformation. Beta conformation organizes the polypeptide chains into the sheets. This is one of the beta strand we can see, and this is in the form of one sheet, and this is the N terminal, and this is the C terminal of the beta sheet. There are 2 types of beta sheets. One is anti-parallel and one is the parallel. In the anti-parallel conformations, the amino acids that are making the hydrogen bonds are facing each other. That's why they are making strong hydrogen bond. While in the anti-parallel beta sheets, the amino acids that are making the hydrogen bond, they are not facing each other. That's why the anti-parallel beta sheets are more stable as compared to the parallel beta sheets. The amino acids that favor the formation of beta sheets are aromatic amino acids like tyrosine, phenylalanine, tryptophan, and some of the branch chain amino acids like the threonine, valine, isoleucine. while the beta sheets are also containing glycine and proline in the turns of the beta sheet. So there are two types of turns in the beta sheets. One is type 1 beta turn and other one is type 2 beta turn. In type 1 beta turn, proline is present somewhere here and the type 2 beta turn is containing glycine somewhere here. In nature, type 1 beta turn are predominantly found as compared to type 2 beta turns. Common secondary structure have characteristic dihedral angles. We can see this is the Ramachandran plot and the phi angles are shown somewhere on the x-axis and the psi angles are shown on the y-axis. We can see if the phi and psi angles are falling somewhere here, the antiparallel beta sheets will be formed. If the phi and psi angles are somewhere here, then the parallel beta sheets will be formed. We can see this area that is containing phi and psi angle that will make the right twisted beta sheets, and this is for the left-handed alpha helices, and this is the region for the phi and psi angle that can make the right-handed alpha helix. We can see for the enzyme pyruvate kinase, the phi and psi angles of all of its amino acids are shown on this Ramachandran plot. We can see there are a lot of dots somewhere here in the deep blue regions, but the phi and psi values for the glycine are not shown because they are falling everywhere in the white region and the blue region because they have too much free rotation. Common secondary structures can be assessed by the circular dichroism. So we already know about the plane polarized light that have one plane and they are falling this way in one plane. If this plane polarized light is rotated right-handed way or the left-handed way, they have a characteristic absorbance value for the alpha helices, beta conformation and the random coil. The difference of absorbance between the left-handed plane polarized light and the right-handed plane polarized light is equal to the circular dichroism. If we take the difference between the absorptivity of the left-handed plane polarized light to the right-handed plane polarized light, this becomes equal to delta E that has been shown on the y-axis. If we draw a graph between the wavelength and the difference of the epsilon value, we come across that the alpha helices have characteristic pattern of the absorbance, and beta sheets are having this kind of absorbance pattern, and the random coil has this kind of absorbance pattern. Protein tertiary and quaternary structures. The protein secondary structure is that structure in which spatial arrangement of all the amino acids that are found in the backbone are explained, while in the tertiary structure, three-dimensional conformation of all the atoms that are present in the backbone and also that are present in the side chain are explained. Amino acids that are far apart in a polypeptide chain, while they fold, they can interact with each other. The location of band is determined by the band-producing amino acid residues like the proline, threonine, serine, and glycine. Polypeptide chains are maintained in their tertiary structure by the use of several weak forces. The protein tertiary and quaternary structure. It is useful to categorize all the proteins in two major categories. One are the fibrous proteins and another one is the globular proteins. The fibrous proteins have the long strands and sheets, while the globular proteins have a spherical globular shape. Fibrous proteins have usually a single type of secondary structure, while the globular proteins often contain several types of secondary structures. Fibrous proteins maintain the sport shape and the external protection, while the many enzymes and the regulatory proteins are globular in nature. Fibrous proteins are adapted for structural function. Alpha keratin, collagen, silk fibrin, they are nicely explaining the relationship between the protein structure and the biological function. Fibrous proteins gives the strength, flexibility in which they occur. All fibrous proteins are insoluble in water. They are containing hydrophobic amino acids both in the interior and on the surface of the protein. The first example of the fibrous protein that we are taking is the alpha keratin. Alpha keratin is evolved for the strength. This protein is found in all mammals. Almost entire weight of the hair, wool, nail, claws, quills, horns, hooves is made up of the alpha keratin. Let's see the structural organization of alpha keratin protein. This is the alpha helix of the alpha keratin protein that is made from the alanine, valine, leucine, isoleucine, methionine, and phenylalanine. Two alpha helices are coiled together to make coiled coil. These two coiled coil are joined together to make the protofilament. So the protofilament is containing 4 alpha helices, and the protofibril is made from the 4 coiled coils, and the protofibril is containing 8 alpha helices, and 4 of the protofibril, that is equal to the 32 alpha helices, is joined together to make the intermediate filament. And we can see these are the cells inside the cross section of the hair that are making the proteins, and this is the alpha helix. These are the coiled coil, then the protofilament, the protofibril, and then the 4 of the protofibrils are joined together to make the intermediate filaments. So this protein is in the range of 44 to 66 kilodalton. Fibrous proteins are adapted for a structural function. We can see the alpha helix, they are cross-linked by the disulfide bonds. This gives them a tough, insoluble, protective structure varying in hardness and flexibility. And the example of these kind of interactions are alpha keratin of the hair, feathers, and the nails. And the beta conformation give a soft, flexible filaments, and they are found in the silk fibrine. And if we see the collagen triple helix, they give the high tensile strength without the stretch, and the collagen of the tendon, bone, and matrix are example of those. Fibrous proteins are adapted for the structural function. So this is very useful example that has been given in this textbook. The person who are having the straight hair, they do have the disulfide bond between the amino acids that are facing each other. If a person has the disulfide bond in the hair of the alpha keratin protein that are not facing each other, that person will have the curly hair. There is a possibility of converting the straight hair into the permanent curled hair and the curled hair into the permanent straight hair. For example, in this case, we want to curl straight hairs. First of all, we will add some reducing agent so that all disulfide bonds are reduced. And then we will curl those hairs and we will apply the oxidizing agents. The new disulfide bonds will be made that are not facing each other, and this will cause the permanent curling of the hair. The second example of the fibrous protein is the collagen. Collagen is found in connective tissues like tendon and cartilages. Collagen is made from the glycine, X-N-amino acid, and proline, or the glycine, N-amino acid, and 4-hydroxyproline. So the collagen is made from the left-handed helical coils. So the collagen has a unique structure that it is containing 3 amino acids in one of its complete turn. For example, this is one complete turn. This is containing not 3.6, but it is containing only 3 amino acids. This is the field model of one of the helix of the collagen molecule. And 3 of such helices are coiled together to make a tight junction and make the one molecule of the collagen. From a top view, these red amino acids are the glycine amino acids. Glycine helps the tight coiling of the helices. And also the glycine provide the point of tight junction between the 3 helices. In this picture, some of the free space is shown in the middle of the helix, but in actual there is no space because the glycine helps the helices to make the tight coiling. So the molecular weight of the collagen molecule is in the range of 300 kilodalton, and it is containing almost 1000 amino acids. So this is the structural organization of collagen. These are the 3 helices coiled tightly with each other, and this is the one molecule of collagen. Different molecules of collagen are joined with each other through the disulfide bonds, and we can see these are the heads of collagen molecules. These collagen filaments are staggered on each other and cross-linked to give the strength. There is a striation of collagen strand that is equal to 64 nanometer. So alignment of the head groups of the every 4th molecule produces a striation. It means that any of the 4 parallel collagen molecules contribute to the formation of striation. For example, this is giving 1 striation, 2nd, 3rd, and 4th. This is contributing the striation, but the 5th one is similar to the first one, and the sixth one is similar to the second one, so they will simply add to the striation of the first and the second head group but will not give another striation. Fibroin is a silk produced by insects and spiders. It is polypeptide chain or beta conformation. So this kind of silk is rich in the alanine and glycine. So the beta sheets are stabilized by the hydrogen bonds, and in between the beta sheets there are van der Waals forces that are maintaining their integrity. So the beta sheets are already in the stretched form. They are not stretchable, but they are soft in nature because of the formation of the weak interaction, the van der Waals forces. We can see here different sheets of the silk fibron. This is one sheet, this is another sheet, this is another sheet, and they are rich in the alanine and the glycine residues. These alanine and glycine are stabilized by weak interaction like the Zeus dipole, etc. This is the colored photograph of scaling electron microscopes. These are the spinnerets of the spider that are excreting the silk. Structural diversity reflects the functional diversity in globular protein. For example, if we do have a protein like human serum albumin of the molecular weight 64.5 kilodalton with a 585 amino acid residues, if this protein is found in the beta conformation, its size will be in the range of 2000 multiplied by 5 angstrom. If it is making the alpha helix, then its size will be in this range. But in nature, this protein is found in the form of globular proteins and its size in the range of 100 into 60 angstrom. Myoglobin provides early clues about the complexity of the globular protein structure. Myoglobin is a relatively small 16.7 kilodalton protein that has an oxygen binding capacity and found in the muscle cells. Its function is to store and to diffuse in the rapidly working tissues. Myoglobin consists of only a single polypeptide chain of 153 amino acids and having only one single heme group. Myoglobin is found in the muscles of diving mammals like whales, seals, porpoise in so abundant quantity that the muscles of these animals are brown. This is the ribbon structure of the myoglobin, and we can see these are the alpha helices. This protein actually consists of all alpha helices, and this is one of the heme group that is shown here. This is the surface contour image of the myoglobin protein with the red heme group shown here. This is again the ribbon model of myoglobin in which the hydrophobic amino acids are shown in yellow and red one is the heme group. And this is the field model in which the hydrophobic amino acids are shown in yellow and this is the heme group. This is the structure of heme group that is containing iron in the middle. This iron is making 4 of its bond with the 4 nitrogen, while 1 of its bond is made with the nitrogen of the histidine and 1 bond is free to be formed with the oxygen. Globular proteins have a variety of tertiary structures. We have taken the example of myoglobin, that is 153 amino acid residue, and it's containing 78% of alpha helices and the beta conformation is 0. Chymotrypsin have 14% alpha helices and the 45% beta conformation. In the same way, another protein that is containing only alpha helices is the cytochrome C. 39% of it is alpha helix and the beta conformation is 0, and the cytochrome C is 104 amino acid in its single polypeptide chain. Protein tertiary structure. Primary structure is containing information only about the amino acid sequences and the covalent bonds are found in the primary sequence. In the secondary structure, we can see folding of alpha helices and beta sheets are random coils are there, and the hydrogen bonds actually stabilize the secondary structure. If we talk about the tertiary structure, 3-dimensional folding of a single polypeptide chain occurs in the tertiary structure. And the disulfide bonds, hydrogen bonds, ionic interactions, van der Waals forces, hydrophobic interactions are all present in the tertiary structure, while the quaternary structure involves only when there is more than one polypeptide chain. Association of multiple polypeptides to form a multimeric protein. Same forces as are found in tertiary structure, they all are stabilizing even the quaternary structure. Protein data bank is a big resource that is containing the three-dimensional structures of the proteins that is very useful in the study of medicine. These are containing the three-dimensional structure. Now we move ourselves towards the determination of the 3D structure of the protein. The first technique that is utilized to find the 3D structure of protein is the X-ray diffraction. The amount of information obtained from the X-ray crystallography depends upon how regular the crystal is. If a protein cannot be crystallized, then its 3D structure cannot be found with the help of X-ray crystallography. In this technique, a crystal is placed between the beam of X-ray and a detector. The spots are created by the diffraction of X-ray beam from each atom of the crystal of a protein. Our electron density map is constructed from the diffraction pattern of the spots. Then a model of structure is obtained from this electron density map. For example, we can see here the spots that are obtained from the diffraction of the X-rays. These are giving the electron density map with the help of some computer-aided programs. Then these electron density maps are utilized by a computer-aided program to make a model of the molecule, and thus the 3D structure of the protein is obtained with the help of some complex softwares. The second technique is nuclear magnetic resonance. In NMR, nuclear spin angular momentum of the atomic nuclei is measured. Only those nuclei that are containing odd number of protons or neutrons or both can give the NMR signal. In this technique, static magnetic field is applied to one type of molecules. Under that static magnetic field, all the nuclei arrange themselves in any of the two positions. One is parallel to the magnetic field, that is the low energy state, and another one is anti-parallel, that is the high energy state. After all the nuclei are arranged, a short pulse of electromagnetic energy is given to them. Under the effect of those short pulses of electromagnetic energy, some of the nuclei will absorb energy and will gain a high energy state. And the absorbed spectrum is characteristic of each of the nuclei. Depending on the absorption spectra, we can identify not only the nuclei but also the chemical environment of that nuclei. And all those signals are averaged, and the computer-aided programs are used to find the three-dimensional structure of the proteins. For example, on the x-axis we can see the proton shift is shown here. The proton found in different environment give a characteristic chemical shift. Chemical shift is the resonance frequency of a nuclei relative to the standard in a magnetic field. Often the position of a chemical shift are diagnostics of the structure of the molecules. In the two-dimensional NMR, we draw the proton chemical shift on the y-axis and proton chemical shift on the x-axis. And we get the absorption, and we get this kind of straight line along the absorption of the two of the proton chemical shifts. Then all these spots are utilized in a computer-aided program and three-dimensional structure is found. In this kind of analysis, nuclear overhauser effect is very important. This effect is the transfer of nuclear spin polarization of one population to another population. Because this topic is out of the scope of this chapter, that's why we are giving only the superficial information. The applications of NMR. An advantage of NMR over the X-ray crystallography is that it does not require the crystallized form of protein. NMR can also be utilized for those proteins that are not crystallized. NMR can also show us the dynamic side of the protein structure. For example, the conformational changes, the protein folding, the protein interaction, and the protein interaction with the other molecules. Globular proteins have a variety of tertiary structures. Myoglobin is one of the examples of the proteins in which only alpha helices are found, but beta sheets are not present. There are many other globular proteins that are containing both the alpha helices and the beta conformation. This is the table once again showing that different proteins that are containing alpha helices and beta sheets in different proportions. Proteins in the folded conformation are containing the hydrophobic amino acid oriented towards the center of the globule, and the hydrophilic amino acids are found on the surface of the globule. The structure of globular proteins are stabilized by the hydrogen bond and the ionic interactions. The 3D structure of a typical globular protein can be considered as alpha helices and beta sheets, and they are connected by the segments. Globular proteins have a variety of tertiary structures. First of all, we will define what are the motifs and what are the domains. The motif is a recognizable folding containing two or more elements of secondary structure, and those secondary structures are connected by the connections. A motif can also be defined as two elements of the secondary structure that also represent a small portion of the protein. For example, a beta alpha beta loop. We can see here this is the beta sheet, and this beta sheet is connected by this connection, and then starts the alpha helix. This one is alpha helix. This alpha helix is connected to another beta sheet through a connection, and this makes beta alpha beta loop. This is containing more than two secondary structure, and this structure is called the motif. A motif can also be an elaborate structure that is containing several protein segments folded together, such as the beta barrel. We can see several beta sheets are folded together to make the beta barrel. This kind of structure also comes under the definition of the motif. Usually a motif cannot function solely, but domains can do. Globular proteins have a variety of tertiary structures. A domain. Let's now define the domain. A domain is a part of polypeptide chain that can independently staple and can also move and can also perform the function. Different domains have distinct functions. For example, they can mediate an enzymatic reaction, they can show binding with small molecules, or they can show the binding with other proteins too. Small proteins usually have only one domain. For example, in this case, we can see polypeptide troponin C protein. This protein is containing two domains, one is shown in the gray and one is shown in the blue. These two domains are joined together and they are almost independently showing the globules of each domain.
Rules for the folding of proteins. Hydrophobic interactions contribute to the stability of proteins. Burial of the hydrophobic amino acids inside the core of the globular proteins require at least two layers of secondary structure. The example is beta alpha beta B loop are suitable for the burial of amino acids. These are creating two such layers. Is more stable when the segment is twisted in a right-handed way. The twist not only influences the arrangement of the beta sheets but also the path of the polypeptide connection. For example, two beta sheets are connected by a crossover strand. In principle, the crossover could be either left-handed or the right-handed, but the most beta sheets are found in right-handed crossover. Right-handed connections are shorter as compared to the left-handed connections, and they are easier to make. The twisting of the beta sheets make a twist in the structure they are making, for example, the beta barrel. And the twisted sheets are making the core of many large structures. Here are some of the examples of the array of beta sheets. We can see typical connections in all beta motifs. For example, in this way, we can see these beta sheets are arranged here in this way, and they are passing above each other. But in the example of crossover connection, we can see this beta sheet is passing through the loop of another beta sheets. For example, one person is standing here and is observing the connection between the two beta strands. This is the first beta sheet and this is the second beta sheet. In order to cross over the second beta sheet, first beta sheet should be turned in a right-handed way. If we see this example, this is the first beta sheet and this is the second beta sheet, and first beta sheet has to cross over the second if it is turned in a left-handed way. And we can see right-handed twist is taking less area as compared to the left-handed twist. So that's why right-handed twists are predominantly found in nature. And these are the twisted beta sheets that are making the core of big structures. Globular proteins have a variety of tertiary structures. We can see here this is the alpha helix and this is the beta sheet and this is the beta sheet. These alpha helices and beta sheets are making the barrel here. You can see here these beta sheets are making the barrel, and some alpha helices are also facilitating this formation of barrel. This is called the alpha beta barrel. Protein motifs are the basis of protein structural classification. We have already seen understanding the complexity of the tertiary structure is facilitated by considering the substructures. Taking this idea, scientists have organized all the proteins in different classes based on their hierarchical levels of the structure. All of these databases rely on the data and information deposited in the protein data bank. In this classification, there are 4 classes of proteins. All alpha proteins that are containing only alpha helices, all beta that are containing all beta sheets, alpha/beta that are containing alpha and beta segments interspaced or alternating, alpha plus beta with alpha and beta regions somewhat segregated. Each class of these contains tens to hundreds of different folding arrangements. Protein motifs are the basis of protein structural classification. These are the examples of proteins that are containing only alpha helices. This black color is showing the identity of the protein, and next one is showing the fold, protein fold. This is the protein superfamily that is showing the family, and this is showing the protein name, and this is showing the species. For example, serum albumin, for example, this identity is containing only alpha helices, and this one ferritin like protein is also containing only alpha helices, and these are the protein segments that are only containing the beta sheets. For example, all these beta sheets, and these are beta sheets connected through these connections, and these are also beta sheets only proteins. And these are the alpha/beta. The alpha and beta sheets are alternatively occurring. For example, we can see this is the alpha helix and this is the beta sheet coming, and then again alpha helix is there, and after some time the beta sheets is there. This is another example in which the alpha helices and beta sheets are alternatively present. These are the alpha plus beta in which an array of one secondary structure is separated from another array of another secondary structure. For example, these are the continuous array of beta sheets, and after this continuous array there is a continuous array of the alpha helices. Some proteins or protein segments are intrinsically disordered. In spite of the decades of research about the understanding of protein structures, many proteins could not be crystallized. Even in some cases, some proteins were crystallized, but sufficient part of those proteins was even disordered. Some proteins lack, there are some cases in which some proteins even lack the ordered structure in solution. Some proteins in the absence of definable structures are performing functions. 1/3 of all human proteins may be unstructured or have significantly unstructured segments. All organisms have proteins that fall in this category. Intrinsically disordered proteins have properties that are distinct from the classical structured proteins. So the presence of charged amino acid in the core of the globular proteins make them disordered. Structural disorder has a high charge density. They may be working in the form of spacer, insulators, linkers, and the large structures. We can see ribbon model of hemoglobin protein that is containing 2 alpha subunits shown in gray and 2 beta subunits shown in blue. And the red ones are the heme group. This is first heme group, this is the second one, this is the third one on the other side, and this is the fourth one. This is the surface contour model of the hemoglobin proteins. Again, these are the alpha subunits and these 2 are beta subunits, and 2 of the heme groups are shown here in the red and 2 heme groups are on the other side. Some proteins or protein segments are intrinsically disordered. This is the 3D structure of very famous protein P53. This is the N terminal and this is the C terminal of this protein. This is the linear structure of this protein shown here. This black line along the linear structure of P53 protein shows the degree of disorderness. If the value is 1, that protein is highly disordered in that region. In nature, actually, the P53 is disordered at its N terminal and its C terminal. Its C terminal is making interaction with different proteins like cyclin A, sirtuins, CBB bromo domain, and S100P protein. Whenever it is acting with the cyclin A, it attains a structured conformation that is suitable for its binding with cyclin A. And with the sirtuin, it attains another structure that is suitable for its binding with sirtuins. And for CBB bromo domain, it attains a different structure, and in the same way it attains a structure that is suitable for the S100B binding. All these structures that are interacting with different proteins are shown with different colors. Protein denaturation and folding. Protein lead a surprisingly precarious existence, as we have seen that native proteins are only marginally stable. Most protein must maintain conformational flexibility to function. The continuous maintenance of the proteins under a given set of conditions is called proteostasis. Loss of protein structure results in the loss of function. In this example, we can see this is one of the mRNA and the ribosome is bound to this mRNA and it is producing nascent polypeptide. This nascent polypeptide can be converted into the folding intermediates, and then these folding intermediates can be converted into the native protein by the use of folding chaperones. In some other cases, these folding intermediates can be converted into the aggregate, and these aggregates are of three types. One is the amorphous aggregates, and another one is the oligomeric, and third one is the amyloid fibrils. The other possibility is that the folding intermediates are converted into the misfolded proteins, and these misfolded proteins are converted again into the aggregates. There is another possibility that this misfolded protein is degraded by ubiquitin proteasome system into smaller peptides and then into amino acid to be recycled. This native protein can also be converted into the misfolded protein. If the temperature of a protein is increased slowly, it maintains its conformation until an abrupt loss of structure is achieved. We take example of two proteins, the ribonuclease A and the apomyoglobin. The x-axis is showing the increase of the temperature and the y-axis showing the percent of the folded protein. Tm is the temperature at which the protein is misfolded or denatured. We can see ribonuclease A is misfolded at a temperature around 58, while the Tm for the apomyoglobin it is around 64 degrees centigrade. In this way, we can know that ribonuclease A is more susceptible towards the increase in the temperature as compared to the apomyoglobin. This is the example of ribonuclease A in which a denaturing agent was added and the percent unfolding was observed. We can see at a concentration of 3.5, the misfolding is occurring almost to 40%. Amino acid sequence determine the tertiary structure of the proteins. The tertiary structure of globular proteins is determined by the amino acid sequence. Amino acid sequence is important. This can be proved by an experiment. Certain globular proteins are denatured by heat, extreme temperature, increase of pH, or some denaturing agent. Whenever this denaturing agent is removed, the native structure is achieved. This is called the denaturation. Ribonuclease A is the example of this case. Whenever we add the urea solution to ribonuclease A, its all disulfide bonds are reduced and it loses its activity and the structure. When this reducing agent was removed, the restoration of its activity was achieved. We can see this example in which ribonuclease A was treated with urea and all its disulfide bonds were reduced, and after the urea was removed, it achieved its structure back. Amino acid sequence determine the tertiary structure. This is a peptide of 56 amino acids. Based on the properties of amino acid, we can know that this segment of amino acids is going to make the linker, beta sheets, or an alpha helix. For example, 8 to 15 amino acids are going to make the linker, and 30 to 37 amino acids are going to make alpha helix, and again we can see 45 to 52 amino acids are going to make linker again. In this case, if we join the 8 to 15 amino acid in the sequence of 1 to 20 amino acids, we can know that these are going to make 2 beta sheets along with 1 linker. If we see this segment, we can know that 28 to 39 amino acids are going to make the linker along with 1 alpha helix, and the amino acid 41 to 56 are going to make 1 linker along with 2 beta sheets. If we join these 2 beta sheets with this alpha helix, we are going to get this structure that is containing the amino acid sequence of 28 to 56. If we join the structure of 1 to 20 amino acid and 28 to 56 amino acid in this format, we are going to get the final structure. In this way, minute detail about the amino acids are going to determine the tertiary structure of the proteins. Polypeptide fold rapidly by stepwise process. In this set of figures, we are going to explain how thermodynamics is going to control the protein folding. So this is one of the mechanisms under which the protein are folded, and these are the thermodynamic consideration of that pathway. This first pathway is represented by this funnel. This funnel is containing the protein folding pathways that are multiple. The protein can be folded by this pathway, and we can see there is no perturbation during the process of the increasing to the decreasing energy of the protein. That's why there is no intermediate involved during this thermodynamic consideration of protein folding. In the second consideration, we can know that protein must reach from this high energy to this lower energy in this funnel, and during this folding they are following multiple pathways, but there are many perturbations during the folding. That's why there are multiple intermediates involved. Here in this case, only a few pathways are involved in the protein folding, and also there is no perturbation, so there is no intermediates involved in this kind of protein folding. In the last kind of thermodynamic consideration of the protein folding, we can know that multiple pathways are involved. Any of the pathway can be followed by the protein during the folding, but there is one strong intermediate is involved, that is shown by this crest. And after passing through this crest, protein achieve the lowest energy level and are finally folded. Some proteins undergo assisted folding. For example, this is a protein that is folded into its folding intermediates. Sometimes this folding intermediates is converted directly into the native protein, but many times it needs the chaperones like HSP70. For example, this is HSP70 that is bound ATP. When the folding intermediates bind with HSP70, it changes its conformation and leaves the protein into its native form. Prokaryotes contain GrowEL tetrameric protein that along with GrowES helps in the folding of the proteins. For example, there are two pairs of GrowEL proteins. We name them GrowEL1 and GrowEL2. For example, this is GrowES1 and this is GrowES2. When the protein needs folding, it goes to the active site of the GrowEL1, and then the GrowES closes this active site. 7 ATPs are joined to the GrowEL1. In the next step, the 7 ATPs that are coming from the previous steps are removed from the GrowEL2. In the next step, the ATPs are hydrolyzed and are converted into ADP, and 7 inorganic phosphates are removed. In the next step, the protein is completely folded inside the GrowEL1 and the new protein comes in contact to the GrowEL2. In the next step, 7 ATPs are bound to the GrowEL2 and the GrowES1 is removed along with the completely folded native proteins. In the next step, 7 ADPs are removed from the GrowEL1 and GrowES2 closes the active site of GrowEL2. In this way, this step continues. Defects in the protein folding provide the molecular basis of wide range of human diseases. We already know that the type 2 diabetes, Alzheimer's disease and Huntington disease are because of the misfolding of the proteins. In some cases, secretory proteins are misfolded, are converted into amyloid fibers. The amyloid fibers are highly ordered and unbranched. The segments are oriented perpendicular to the axis of the fiber. We can see the example. This is the native protein. In the case of misfolding or partially unfolding, sometimes this protein is denatured. But in other cases, the alpha helices are converted into beta sheets and they are arranged in the form of amyloids. This is the amyloid fibers and this is the amyloid beta peptide that is converted finally into the amyloid fibrils. Deaths by the misfolding, the prion disease. We see here the molecular mechanism of the prion disease. This is the 3D structure of the prion protein that is containing 1, 2, 3 alpha helices and 1 or 2 beta sheets. When this protein is misfolded, it is containing only 2 alpha helices and multiple beta sheets we can see. These multiple beta sheets come into the amyloid form and are aggregated. This makes the molecular reasoning of the prion disease. So thanks for watching. This is the end of chapter 4.